We're going to talk about viruses, which I think are some of the most interesting of the microorganisms that we're going to see this semester. Recall that we introduced the two different general types of viruses in Chapter 1. We have bacteriophages and we have animal viruses. Viruses are also incredibly small compared to the other organisms we've talked about so far. They're going to require an electron microscope to see them. And we kind of mentioned that there was a controversy over whether they're alive or not. So we're going to talk about some of those characteristics. Viruses are incredibly compact. When we say they're acellular, they don't have the membranes and the organelles that other species have. However, they do have some similar substances we're going to talk about. For example, nucleic acids. So viruses do have either DNA or RNA. But they don't have them packaged the way that bacteria or eukaryotic cells have and other organelles. They're also obligate intracellular parasites, which means they require a host cell. So there's going to be two life stages, if you will. What we call active, they're not necessarily alive, but once they get inside of a host cell, they're really just in their nucleic acid form, either DNA or RNA. And we would call them the intracellular state. When they're inactive or inert, it's because they're outside of a host cell. So when they're extracellular, we use the word virion. So virion is the form of the virus that gets transmitted typically. So the viruses in the cold and flu season that get left behind on doorknobs, then you touch it and then you introduce it to your mucous membranes, it would be in the virion form. So one of the reasons why it's controversial about whether they're live or not is because some of the characteristics we use to determine whether something is live or not is whether they can grow and reproduce independently. Viruses require host cells to use their organelles, their enzymes, in order to copy themselves. They also can't metabolize on their own, so they have no feeding mechanism or anything like that. So that's one of the things that sort of puts it in the column of not being a live organism. Typically, live things don't respond to their environment as well. And so one of the things that's fairly recently understood about cold viruses in particular is that their protective covering, what's called a capsid, which we're about to meet, actually gets thicker during the cold weather. So this is one of those controversial kind of factoids that make viruses sort of ride this line between are they live or dead. So all viruses have at least this basic structure, which you have the nucleic acid that then's protected by a protein coat called the capsid. And you'll see at each of the corners of this particular kind of capsid, this is a very geometric shape, what we call a icosahedral capsid. But you'll see at each of those corners, what they've named as spike here are basically glycoproteins that are going to be used by this virus to as what we call a host recognition site. That's the little thing that binds to the host cell and allows it to infect a certain host cell. Some viruses have that original anatomy that we just looked at, but then they're surrounded by yet another layer called an envelope. And this envelope is basically made of phospholipid bilayers, just like a cell membrane, but they don't actually make that membrane. They steal it from the host. So generally we have two types of viral structure, what we call naked viruses, also sometimes called non-enveloped viruses, or enveloped viruses. So you can see if it's an enveloped virus, those spikes or those glycoproteins that they use as affinity sites are going to be on the envelope rather than on the capsid, because that's where the surface is that would come in contact with the host cell. So viruses can then further be broken down by the genetic material that they contain. So for example, they can either have DNA or RNA. They would never have both forms of that genetic material. But they have much more variation in how that DNA and RNA is put together than most cells. We'll talk about that. How that DNA is organized, it can either be linear, like ours, right? Ours are in a long double helix chromosomes, which are just long segments, multiple segments like ours. We have chromosomes. Or it can be very small segments of DNA that are just singular in little circles, much like a plasmid that you would see in a bacteria. So lots of variety in their genetic compositions. And so when it comes to the possible forms of those genetic material, they can either be in the form of what we call double-stranded DNA, which looks just like our cells. It could be single-stranded DNA, which again mimics what ours look like when our DNA has been unzipped for the purpose of replication or protein synthesis. They can come in the form of double-stranded RNA, which is unlike what our cells have, right? Our cells only carry single-stranded RNA. So we're going to look at the combinations of these. Now keep in mind when viruses are put in taxonomic groups, they don't fossilize very well, so we generally can't look at their genetic evolution the way that we do other organisms. So they really don't have a species designation. A lot of viruses just use an English vernacular, a common name, so to speak. So think rabies virus. We do have viruses that are grouped into at least 350 genera. So we'll get them down to the genera level. But then beyond that, there's usually not a species designation. So again, they try to put them in the same kind of taxonomic groups where we have orders, families, classes, and those things, but they stop at the genera level. So here's a good sort of summary table of a couple of examples of DNA viruses versus RNA viruses. We're going to meet each of these types of diseases along the way through the semester, but here's just sort of an introduction to some of those. When it comes to their genetic material, their genomes tend to be very small compared to other organisms. So for example, the human cell contains roughly a 40,000 genes within their genome. Now, we know that number is continuously going up as we continue the Human Genome Project and research. Since then, we know that number is drastically underestimating the possible genes in a human cell, but this is just for reference, for relative sizes. Bacteria, the largest bacteria, contain on the order of about 4,000 genes. Some of the smaller bacteria, maybe 1,000 genes. Then we cross over into the viruses. So herpes viruses are some of the largest viruses, and they have, on the order of 100 genes or more. But there is a small group of viruses that have, up to 1,000 genes. So the largest viruses sort of mimic that lowest or smallest bacterial cells. But herpes viruses, this is a huge family of viruses, by the way. So we'll talk a lot about those this semester. Bacteriophages are very small, very compact, very simple organisms that maybe have, in the 30 to 56 genes. And then hepatitis virus, that's one of the simplest viruses we know. It only has 4 genes. So they literally only carry the information they absolutely need. They're very light packers, so to speak. They we'll talk about what those genes would represent to each of these viruses. Now, viruses can also be categorized by who they infect, right? So the only hosts that they can infect are those that they have affinity for. So what's kind of ironic is that most viruses have to be invited in by the host cell. So we don't have receptors on our helper T cells for HIV virus. We just have CD4 receptors that are functional receptors for other parts of the immune system function that HIV just happens to fit into because they have these affinity between these surface markers and their host cell. Now, in some cases, that affinity is going to have a much broader fit, so to speak. So if it's a virus that is a generalist, it's going to be one that can infect many types of cells and sometimes many different hosts. So for example, the polio virus can actually infect, it gets into the body through the digestive system, but then once it gets into the body, its affinity has a preference for the nervous system cells. So that would be a generalist. It has more than one target. The rabies virus, why it has so many mammalian targets is because, or hosts, I should say, is because its affinity is for acetylcholine receptors. So think about where acetylcholine receptors are on every muscle in every mammal. So now that doesn't mean every mammal that encounters the rabies virus is capable of passing it on, and we'll talk about that when we get to the nervous system diseases. But it can in fact infect multiple species, so that would be a generalist. Specialists, on the other hand, are those that can only affect one type of cell in one type of host. So specialists would include things like HIV. Now, the more research we do on HIV, we realize once the infection has progressed to a certain degree, that virus can actually infect other types of cells within the human body. What we do think of HIV as a specialist. Hepatitis as well, right? This is not a disease you can get from other animals. It's strictly a human disease, and it only affects the liver cells, thus the name hepatitis. The mumps virus also only affects human beings, and it has a special affinity for the salivary gland cells, which is why when you have a mumps infection, you look sort of like a gopher because those salivary glands swell up. Now, all bacteriophages are specialists. They have an incredible affinity, one bacteriophage to one type of bacteria. So we'll talk about those a little bit as well. Remember, all viruses have capsids to protect that nucleic acid. And so capsids are going to be these little protein coats. They're made of proteins called capsomeres. And they may have one single type of capsomere. Those are going to be very simple capsids, or they might have multiple types combined. So we mentioned that sort of geometric shape one. The faces of that icosahedron would be one type of capsomere, and then the corners would be a separate type of capsomere. So those proteins then are put together in different varieties. So if it's a helical, think of like a hollow tube essentially, those would be helical capsids. One subtype of a helical capsid would be the filoviruses like Ebola. We'll see that some examples of each of these here shortly. Icosahedron, we mentioned those are going to be very geometrically shaped capsids. And complex capsids, and these are going to be where you have a special layering, so there's multiple layers of capsids. Now keep in mind, this capsid is really what makes the virus, it's the bulk of the virus, and it's what gives the viruses their general shape. So some examples, in fact, this is the first virus ever discovered, was called the tobacco mosaic virus. It's a really large virus, but these helical viruses, or helical capsids, are made of a single type of capsomere, and they're literally just layered together until they make this hollow tube. And so these are really long, really symmetrical type of capsids, and the DNA would just be inside of that hollow tube. Helical capsids include the filoviruses. So, for example, the Ebola virus and the Marburg virus, both of these are filoviruses. We'll talk about these diseases as well when we get farther down in the semester. But one of the most characteristic features of this is they are, in fact, the hollow tubes, but they're flexible. The true helical capsids are not flexible. They will literally sort of break apart into pieces, but the filoviruses are very flexible, but they do have this very characteristic thing at the end, what they call the shepherd's hook. So that's usually the thing that the pathologists, if they see that, they know they're in trouble, because neither of these, any of the filoviruses that we know of, are really dangerous diseases. So we'll talk about those later in the semester. Here again are the icosahedrals. Most of the animal viruses are going to look like this. The prefix icosa, by the way, just means 20. And so when you look at those little geometric shapes, they have 20 faces on that capsid. And then again, if you look down here at the naked versus enveloped, the naked viruses, they would put those glycoproteins, those host recognition sites, right there on those corners of the icosahedron. Complex capsids are going to be those that sometimes is just an odd shape, or it's going to have multiple layering. So for example, the rabies virus, it's famous, it's easy to identify under the microscope because it looks like a bullet, as you can see. It does, in fact, have an envelope outside of that capsid as well. So that's what you're seeing there is the envelope with all the little glycoproteins on the surface. The smallpox virus is a complex capsid. It actually has sort of roughly oval-shaped capsid, and then it has a second layer of capsid that's sort of wrapped around it like a little hot dog bun. So that red center that you're seeing there is actually sort of the gap on the inside of the outer capsid. But that by having multiple layers of capsids, it makes it very tolerant of environmental conditions, which is one of the reasons why that virus, if you think about historical context, that the smallpox virus was brought to the New World by Columbus and other travelers that on the blankets of people that had smallpox back in Europe, and they brought that virus over because the virus was still viable on those blankets because of this complex capsid that's very, very hardy. Bacteriophages are in fact complex capsid, excuse me. The head portion is icosahedral. The tail portion is a helical. So they're not always layered on top of each other. They just include more than one shape. Now, if a virus is enveloped, it's going to be made of phospholipids, and it's going to have to steal it. Acquired is a nice way of saying stealing or hijacking that phospholipid from the host cell. Now, typically it's going to be taken from the cell membrane as the virus is shed from the host cell. But think about all those membrane-bound organelles that we talked about in chapter 4. We've got multiple places in that host cell that the virus may actually steal that envelope from. It may come from the nuclear envelope. It may come from the Golgi or the ER. It wouldn't come from the mitochondria because that is not a movable membrane, and the viruses don't ever get into the mitochondria. But think about those enveloped, those membrane-bound organelles. Those are all possible sources of this envelope. And so that phospholipid bilayer is taken or stolen from the host. The proteins that are embedded on it are going to, again, we keep mentioning this host recognition site. If those surface proteins don't match the target, then they're not going to be able to infect that cell. And so when we mention those host recognition sites, if they're called glycoproteins, they're probably made by the host cell and they were stolen along with the envelope. If they call them spikes, they're probably something that the virus was carrying the DNA for that shape, and they posted it out on that envelope themselves. And so we'll talk about some of those, the differences between a spike and a glycoprotein. They're both basically made of glycoproteins, but they sometimes use that term to just differentiate where the DNA code for that surface protein came from. So here's some examples of viral envelopes. So again, the capsid can be either helical or icosahedral, but then the envelope was really going to give these guys their shape. And so this envelope, because it's not a membrane that the virus produced, it can't maintain it, it can't repair it. It's roughly spherical, but it can't, and it's flexible, so it's not something that the virus has any control over its shape, essentially. But it really helps it function sort of like a Trojan horse, right? Obviously, if it's made of phospholipids, and especially if it's coated in glycoproteins that came from the host cell, you can see why the immune system has a real hard time going after these, because if they look like something that belongs, they're going to get ignored. Another type would be here. So this one would have the icosahedral capsid, and then the envelope on the surface here. So again, here they're showing you those different shapes of glycoproteins, some of which are sort of spike-shaped. Those would probably be those that are produced by the virus rather than the host. So again, just to kind of give you a relative size comparison here, you can see there's a human cell, the red blood cell. The bacteria would be much smaller than that. And then if you zoom in, you're going to get down here to these viral shapes and sizes. So generally, viruses are going to fit somewhere between 10 nanometers and 450 nanometers. And just to give you some relative size here, you could fit about 2,000 phages in a bacterial host cell. You could fit about 50 million polioviruses in a human cell. So incredibly different size classes here, but you can see some of the largest viruses are things like the tobacco mosaic virus and the smallpox virus. They actually in 2003 discovered a virus called the Mimi virus and it was so large that it mimicked bacteria, it took until 2003 before they realized when they did genetic studies that it's actually a virus and not a bacteria. So wide range of sizes, but think incredibly small, ultra microscopic. So let's start with the bacteriophages. We're going to talk a little bit about their life cycle, how they're put together. But we said these guys are specialists, so they're going to have generally a head area, which is the capsid that contains the nucleic acid. They're going to have a tail portion, which is the helical capsid plus these tail fibers. And it's at the end of those tail fibers. Most people look at this and think that those little tail fibers are like legs that would help them move around, but they're non-motile. But they do contain those affinity sites out there at the end of those tail fibers. So when they land on a host cell, those tail fibers have to have affinity. Now at the base of that tail, you can also see what's called the base plate. And in that base plate, they may contain enzymes that when this organism lands on its bacterial host cell, it will sort of squat down, if you want to think of it that way, and that base plate would then come in contact with the cell wall of the bacteria that it's trying to infect, and those enzymes would start to dissolve away the cell wall, and then the nucleic acid that's up there in that head would literally be forced through, kind of like a hypodermic needle, into the bacterial host cell. And so bacteriophages basically bust their way in. They use brute force to get into their host cells because of this anatomy. And when they replicate, once they get inside their host cell, they're going to have two forms of replication, or two different life cycles, if you will. The one group is going to be called the lytic phages, and they sometimes are called virulent phages because virulence means dangerous, right? These guys are literally going to kill their host cells. After they've replicated, they're going to bust out and kill the host on the way out. And so that's why they're called lytic, because they cause lysis in the host cell. The other family are called lysogenic phages, and these guys sometimes also get the nickname temperate phages. So I always look at the root word there, temper, as in tempered glass, right? So you think about your vehicle, your windshield, and all the glass in your vehicle is what we call tempered, which means when it shatters, it doesn't break into very sharp pieces. It breaks into less dangerous, sort of blunt-edged pieces. And so these guys get that name because they don't outright kill their host. They actually incorporate themselves into the host DNA, and basically they can stay there forever, or they can just temporarily be hidden out in that host DNA. And so once a lysogenic phage has found a host and has inserted itself permanently or temporarily into the host DNA, it's now called a prophage. So this is what the lytic replication cycle would look like. First, you have to attach or adhere. That requires affinity between the host and the virus. Then entry, we just mentioned, the primary method of bacteriophages is just brute force. It's going to force that nucleic acid right through the cell wall using the pressure that's built up in that head. And then the capsid that's left over really just becomes debris. It's going to float away. It's not going to get used again. It just becomes junk, so to speak. But once that nucleic acid has entered the bacterial host, the first thing it does is engage some enzymes that degrade the bacterial chromosome. And so it's literally going to chop up the host cell DNA. So number one, the host cell DNA can no longer function, but it's also going to use those pieces, right? DNA is DNA. The virus is going to have to copy itself. So where's it going to get the new nucleic acid that it wants to look like on its way out? It's going to literally steal it from the host. And so to do that, it needs to break down the host chromosome. Once it's done that, it will start to synthesize all of its components. It'll have to make new capsid proteins, it'll have to make new tail fibers, it'll have to make copies of its DNA, things like that, and then it will have to assemble. And the assembly phase is really the section of the part of this life cycle that we know very little about, especially because of that pressure in the head. It's really interesting that they can look at the assembly stage and they can find all the individual components still free floating, and then they come back later and they can see that DNA has sort of been forced up into that head, and somehow they sort of pump up the pressure in there. There's some new research that shows that DNA gets spooled in a very particular way or sort of coiled up, and that helps to increase the pressure within that head. Lysogenic phages, on the other hand, are going to, again, insert themselves or incorporate themselves into the host DNA. And so once they've done that, they're going to be called a prophage. The host itself has now a different name. If it is harboring the enemy DNA in its chromosome, it's going to be called a lysogenic cell. So you can kind of think of this host as a sort of complicit, right? It has allowed for this virus to insert itself. And it might be strange that they're allowing that to happen, but remember, bacteria are accustomed to sharing plasmids. So they're not averse to taking on new DNA because of their own physiology. So that's part of why this is possible with this family of viruses. Once they've inserted themselves in the host, they may or may not do anything, right? And so if they're just sort of hiding out in the host DNA, they may be referred to as silent viruses. And that's what they mean by a lysogenic virus. It's basically a silent, it's just waiting out its life cycle. Now, if something inducts it or reactivates it, if it goes through a process called induction, it then would come back to life, so to speak, and start copying itself. So you can kind of think of those things that would trigger, if we were talking about human infections like herpes, right? Herpes is forever. That virus is inserted in your DNA and it's in your body forever. Now, it doesn't necessarily do anything, but there are certain conditions. So, for example, cold sores, right? We think of cold sores, fever blisters. Those are herpes viruses that are just waiting out in the nerve cells. And a really severe sunburn, fever, stress, sometimes a cold, right? We call it cold sores because when your body is stressed, those viruses reactivate or get inducted, and then they start copying themselves. And once a virus, a lysogenic phage, has gone through induction, it basically becomes a lytic virus. It's going to then kill the host as it copies itself. So here's what that life cycle looks like. So again, it looks very much like the lytic stage to start with, but once it enters, rather than destroying the host cell, or chromosome, it just uses the enzymes to break that chromosome apart and insert itself. And one of the big advantages of lysogeny is that now it's inserted in its chromosome of the host. Every time that host cell copies itself, it will inadvertently copy that virus as well. And so it sort of gets a free ride. So it's copying itself every time that the host cell divides. And remember, bacteria have incredibly fast regeneration rates. So we're talking every 30 minutes, E. coli might double in size population-wise. So it's a big advantage to the virus to sort of just tag along in that process. And then if induction occurs, then it converts into actually replicating the virus through the synthesis stage, assembly stage where it puts it all together, and then release. At the final stage of the lytic cycle is to be released. And this is where those new fully formed virions are now then free floating and they can go find a new host cell. We've got an animation here that walks through these life cycles. Let me pull this up. And so why do we care about phages if they can only infect bacteria? Well, we care because some bacteria that are not pathogenic to us become pathogenic once they have been infected themselves. And we call those bacteria lysogenically converted. So when lysogenic conversion occurs, think about what has just happened. That bacteria now has DNA it did not have before, which means it has the ability to make proteins it could not make before. And so when a prophage is present, some of the genes that they take on are actually for toxins. So, for example, there are several diseases that rely on toxins for doing the damage that essentially causes the damage to the human body, such as diphtheria, right? Diphtheria is a toxin-based disease. If that bacteria was not infected by a bacteriophage, it would not possibly make the toxin that causes the disease. Same thing goes for cholera. Botulism, right? So the botulinum toxin is actually a lysogenically converted gene. There are certain strains of Streptococcus that, if they are lysogenically converted, can lead to scarlet fever, for example. So there's a reason not everybody that gets strep throat ends up with scarlet fever. right? It depends on whether the virus or the bacteria that you've been exposed to was previously infected by the virus, the bacteriophage. So this is why we really are concerned with bacteriophages and understanding how they contribute to human disease. Even if they can't do it directly, they're very dangerous in some cases. What about animal viruses? Well, if we wanted to copy an animal virus, it's generally going to look the same. The main difference is how they enter and how they exit, right? They're not going to just bust in because we don't have cell walls. So the presence of that envelope makes it much more subtle, right? They can just fuse that envelope with the cell membrane and have direct access to the host cell without damaging it. Once they get in, some things that are going to differ are the fact that there are organelles, there are different enzymes and things in that eukaryotic cell than there would be in a bacteria. And of course, how they get in, how they get out is going to look very different because there is no cell wall in our animal cells. So primarily it's about that whole brute force thing. It's not going to need brute force because there's no cell wall to have to get past. Just like in bacteriophages, though, the attachment or absorption stage, they have to have affinity. If they don't have a matching shape, they won't be able to infect that host cell. So this right here, understanding that what makes those receptors and what makes those glycoproteins is DNA. So this right here is the reason why not everyone is susceptible to the same diseases. Right? If your receptors don't happen to have affinity for this year's flu virus, then it can't infect you regardless. So the whole idea of, this year's vaccine didn't work for me or didn't work for a lot of people, it has everything to do with geometry of these proteins. And so keep that in mind when we start talking about the immune system and things like that. It has everything to do with affinity between surface markers of the virus and the receptors on the host cell. How do they get in? Depends on what kind of virus they are. So if they're a naked virus, remember we said those glycoprotein host recognition sites are going to be found on the corners of that capsid. They would have to then bind to receptors on the host. And then when the host cell has enough of those receptors bound, it's literally going to sort of invite the whatever's bound to those receptors in. In this case, we call direct penetration. The only thing that enters is the nucleic acid. That capsid then, again, just stays on the surface of the host cell and becomes debris. Envelope viruses have a couple of ways they can get in. Same thing, they still have to have affinity with their surface markers, but once they have bound, the two membranes, right, the cell membrane of the host cell and the membrane of the envelope will literally just fuse together. So we call this membrane fusion. And in this case, the entire capsid and nucleic acid would get inside the cell. And this creates a problem actually because we don't understand really for most viruses how they do what's called uncoating, right? That capsid is separating that nucleic acid from any of the enzymes in the host cell that might be able to copy it. So it has to uncoat in order to be able to then replicate and infect the cell. The third option might be endocytosis, or you could think of this as phagocytosis, right? That if it binds to the right kind of receptors, then the phagocyte or the cell would just literally sort of consume it. And once it gets inside, again, it relies on the fact that membranes fuse beautifully together. Once they fuse together, then the virus somehow uncoats. And this is the one thing we understand about uncoating in some viruses is the fact that they actually take advantage of lysozymes. So remember we talked about phagocytosis. What would happen is during this stage here, once it's been ingested, that membrane-bound vesicle would then fuse with a lysosome, and the lysosomes would actually be responsible for doing that uncoating. So some viruses actually take advantage of phagocytosis to get out of their own trap of capsid. Now, once they get in, it really depends on which form of nucleic acid the virus is made of, to determine whether the host or the virus is going to be responsible for copying that virus. So the enzymes are going to be responsible for either unzipping the DNA, replicating the DNA, things like that. And so you have to think about two things. If you're going to make enzymes, if you're going to make proteins in general, like capsomeres. If you're going to make a capsid, you're going to have to make protein. If you're making protein, you've got to use the ribosomes, right? And ribosomes only use messenger RNA. So you've got to get whatever type of nucleic acid the virus came in looking like into something that looks like messenger RNA in order for them to copy themselves, right? There is protein synthesis involved in viral replication. So when you think about what we're doing here, these two things the two questions we're asking for here are, depending on what kind of nucleic acid form you come in looking like, the more similar you look like the host, the more likely you're going to use host enzymes for both of these steps, right, for copying yourself. So how is messenger RNA going to be synthesized, and what will serve as the template, right? So obviously you have to make proteins, and you have to make more of the nucleic acid that you came in with, right? If you're making more viruses, you need to look just like you did on the way in. And so when we say template here, remember that when we look at nucleic acids, they are, if it's a double strand, they're not copies of each other, right? They're not Xerox copies of each other. When we talk about nucleic acids, we're talking about ACs, Ts, and Gs, right? Adenine, cytosine, guanine, and thymine. And uracil, if you're talking about RNA. Remember that cytosine sits across from guanine. Adenine sits across from thymine. These are what we call complementary strands, right? They're not identical. So when we talk about a template, if you are a double-stranded DNA virus, you would use the opposite strand as your template, right? So you can kind of think of this as how you're going to make a copy of yourself. You're going to need something that looks like your exact opposite in order to do that. So double-stranded DNA viruses, they come in looking just like something that belongs. So they're going to use the host enzymes for unzipping that double strand of DNA. In fact, we call that a helicase. Remember, it unwinds the helix and then unzips the double strands. It can use host enzymes to then transcribe that DNA into messenger RNA, and then use the RNA polymerase to then translate those proteins at the ribosomes. So they've really got it easy, so to speak. And then they're going to use templates. Obviously, we do have enzymes that know how to make DNA from DNA. They would just use the opposite strand as a template, and they've got it pretty easy. Now keep in mind, because they are DNA viruses, and especially double-stranded DNA, because they look like something that belongs, they can also insert themselves into the host DNA and stay forever, right? So there's a reason why we say herpes is forever. Once that virus is in your body, it's there forever. Same thing goes for the papillomaviruses, which is going to be important when we talk about cancer. That those human papillomaviruses that can cause cervical cancer, it's because they can insert themselves in our host DNA and stay there forever. And whether they cause cancer or not is something else that we'll talk about here in a minute. But these are some examples of double-stranded DNA viruses. Single-stranded DNA viruses also have life pretty easy because isn't on occasion our DNA unzipped? So yes, we do have enzymes that know how to copy and deal with single-stranded DNA. And so again, they're going to have everything identical. Examples of single-stranded DNA viruses are something like the parvo viruses. Now we get into the RNA viruses. So single-stranded RNA viruses, we're going to have two flavors. We're going to have positive and negative, and the positive strands are sometimes called sense strands, and I like to think of that as the sense strand positive single-stranded RNA viruses are those that the ribosomes can make sense of, right? So because positive single-stranded RNA virus is identical to messenger RNA, it can go directly to the ribosome and immediately start making enzymes, right? It's going to use the host enzymes. It's coming in looking just like that, that is required. But because we don't have double-stranded RNA, the template that would be used to make more of that positive strand would have to look like the complementary strand to that positive strand, and we would call that negative single-stranded RNA. And so because we never make RNA from RNA, we would have to, that virus would have to come in pre-loaded with an enzyme that it made in its last life cycle which we call viral RNA polymerase, right? If you want to make RNA, you use an RNA polymerase. We make RNA from DNA, not RNA. So they would have to bring their own copy in order to copy themselves. Some examples here like poliovirus and the picorna viruses. So remember, pico is like 10 to the minus 12. It's a prefix for the metric system. Very, very small viruses, and they're made of RNA. So there's the name picorna virus. There's a special subclass of positive single-stranded RNA viruses called the retroviruses. Again, because they're positive stranded, they are sense viruses, which means they go straight to the ribosome and they can start making enzymes using host enzymes, or start making proteins using host enzymes. They will also need a viral RNA polymerase in order to make that opposite strand if they want to use that as a template. But where the name retrovirus comes in, right? Retro, you can think of going back. What's going back here is that RNA strand could be turned in reverse into DNA, right? When we do transcription, we're going from DNA to RNA. These viruses have an enzyme that can go the other way. And so if you have a reverse transcriptase enzyme, now you can go from being something like a temporary molecule like RNA, and you can convert yourself into something that is a permanent molecule like DNA. Right? And so this is similar to what we did in the bacteriophages called lysogeny. Now, these animal viruses that are retroviruses can insert themselves into the host and stay there forever. And now they can use that DNA as a template. They don't necessarily need to make the RNA template, but they will do that as well. They can do both, right? And so here the example, most people have heard the term retrovirus with HIV. But this term oncovirus, we're going to talk about cancer and viruses here in a minute. That's just a general family of viruses that we know are capable of causing cancer. So that prefix onco means mass. That's a prefix that we use for a lot of terms associated with cancer. And again, it's because DNA is a more permanent molecule. So retroviruses are going to be a special class of positive single-stranded RNA viruses. Negative single-stranded RNA viruses are sometimes called antisense because when they get into the host cell, the ribosome can't do anything with them until they've been converted to the positive version of that molecule. And so this one, again, these guys are very different than what is found in our cells. They're going to have to bring pre-made RNA-dependent RNA transcriptase. I know it's a big, long mouthful, but basically, again, what you're trying to do is make RNA from RNA, which we don't have enzymes to do that. And so first they have to transcribe that positive single-stranded RNA, and then once they have that, they can use the host enzymes at the ribosomes to make proteins. They will need viral enzymes, however, to make templates, because again, we don't make RNA from RNA. So they can use that positive strand that they made as either the molecule that goes to the ribosome to make proteins, or we can use that same strand as a template to just make more of the negative strand RNA when they're assembling themselves. This group includes things like the rabies virus, flu virus, Ebola, and Marburg viruses, which are those filoviruses we just saw. But keep in mind why flu virus is problematic is because it is an RNA virus. Not all RNA viruses mutate quite readily, but this one does. So, for example, the reason why we need a new vaccine every year is because flu viruses are RNA viruses. They're much more likely to make mistakes, and those mutations then allow the virus to change constantly. So we'll talk a little bit more about that idea as we get further on in the semester. Double-stranded RNA viruses, also, this is something that our cells never have. And so they would initially have to bring in a pre-made enzyme. If they don't come with a viral enzyme that knows how to unzip double-stranded RNA, they're sort of dead in the water. They can't replicate at all unless they come with a pre-existing enzyme that can unzip them. Once they've unzipped, they're going to make that positive strand from the negative and the negative from the positive. They have their own sort of template, but they're going to need viral enzymes to do that because, again, we don't make RNA from RNA. Once they've made the positive strand, they can use the host at the ribosomes to make proteins. But again, those viral enzymes are going to be needed to make those complementary copies of the nucleic acids themselves. So this includes a group that we just refer to as the REO viruses or the REO viruses. This stands for respiratory, respiratory enteric orphan viruses. And so these are just sort of a very general group of viruses that have similar symptoms. They're either respiratory or digestive symptoms. We rarely diagnose them. They affect babies very oftentimes. So, but this is a double-stranded RNA virus family, and again, they don't look like anything that belongs in the human cell, and so they're going to have to, or in animal cells in general, they're going to have to bring pre-existing viral enzymes in order to copy themselves. So once they've assembled, as they release from the envelope, from the host cell, some of them won't release immediately, right? So some are going to insert themselves into the host DNA and become what we call latent infection. This would be equivalent to what we call in phages lysogeny. So we do use those two different terms to specify bacteriophages versus animal viruses. So if someone has a latent infection, it means the virus is present. It's just not currently actively copying and causing symptoms. And so just like in the lysogeny, we called those prophages. Once the viral DNA of an animal virus has inserted into its host cell, it would be called a provirus. Herpes viruses, for example, right? Herpes viruses cause cold sores, also cause genital herpes, those things. The same idea that brings a lysogenic virus out of lysogeny would be induction. And so think about those things we just mentioned. Cold sores get triggered or inducted by stress or fever, things like that. HIV is also one of those examples where it can be just latent. It can be in the host cells, but not doing anything for decades. Chickenpox, right? As a child, you had chickenpox. That virus is permanently installed in your body. You may or may not ever see shingles as an adult, but when we do, it's usually because they got inducted, right? Very oftentimes, shingles occurs in adults that are stressed or just age. Your immune system takes a hit for some reason, and that shingles is basically a reactivation of the same virus that was latent in your body. Latency and persistence are two different characteristics. Persistent infections are those where the virus is in the host and they are just continuously releasing virions from live host cells, and they never really kill the host cell. And so you get this gradual buildup, and so you can get enough of them built up in the body that can be fatal. In some cases, things like measles and rubella infections, if you didn't have the vaccine against these two diseases, those viruses can build up to a persistent level to the point where it can become a very serious version of encephalitis. So there's a reason why we vaccinate against these two diseases, even though they're very mild infections. If you're a child that gets measles or rubella, you probably survive. But in some people, that virus can cause lifelong problems, right? Encephalitis, in some cases, certain forms of measles can lead to permanent damage to the reproductive system, which you won't see until you're an adult. So again, there's reasons we'll talk about certain diseases, why we have vaccines against some, but not all diseases. This is one of those characteristics for these two diseases, why they developed a vaccine. So again, if you're an enveloped virus, you have multiple ways to leave the host cell without killing it. And one of the ways, what's sometimes referred to as shedding of virus, is also technically known as budding, right? So as it leaves, it's going to be targeted to a certain part of the cell membrane where those glycoproteins are waiting on the surface, so that when it sort of pinches off and takes part of that membrane with it, it's now coated with those glycoproteins. And so this, persistent infections would look like this. The host cell is still alive, it's just pumping out virus. If it's a naked virus, on the other hand, it can leave in one of two ways. Exocytosis, which looks a lot like that budding that we just saw, where it doesn't kill the host cell, but in many times it will actually kill the host cell on the way out, much like lytic phages did. And so in those cases, that's sometimes the mechanism behind why you see lesions with viral infections, right? So some of the STDs we're going to talk about, herpes, for example, it leaves behind these lesions because you're actually killing the host cells that the viruses have replicated in. Certain types of infections that affect the respiratory system, why it leads to a lot of damage to the respiratory system is because it actually kills those cells lining the air sacs of the lungs. So this is a good sort of compare, contrast, just some basic differences between the bacteriophages and the animal viruses. So this is a good little summary to study. So let's talk about how viruses are involved in cancer. We mentioned the fact that they can insert themselves sometimes in the host DNA. When they do that, we call that transformation. So when the DNA has been transformed because of a virus that's present, it can lead to cancer. So this number keeps going up, by the way. About 13% of cancers are now confirmed to be associated with certain viruses that get into the DNA. And so we call those oncoviruses or oncogenic viruses. Think about what cancer is. Cancer is uncontrolled cell growth, right? At its simplest form, that is what cancer is. And so if DNA controls the growth of cells, there are certain genes that control whether those cells are growing or are inhibited from growing. Viruses that come in carrying what they call oncogenes may cause cancer. The oncogenes that are naturally present in our DNA may just be stimulated if the virus parks itself right next to it, so to speak. But in most cases, what's happening with viruses is that they come in and they interfere with the genes that are known as tumor repression genes. So all animal cells, if you're a multicellular organism like a human being, your cells going through development, we go from a single zygote to trillions of cells in the mature full organism, right? And so when you're going through embryology, you have thousands of cells that are replicating constantly. And to get them to their mature adult size in an embryo, it's a lot easier for all of the cells in that developing embryo to sort of be on full go mode, right? So they are just, the genes are triggered to say grow, grow, grow. And the thing that stops that growth is typically triggered by something called contact inhibition. So let's say you're a liver cell, and when the liver has reached its mature size, those cells that are making up the liver will literally be bulging to their full size, and they're putting pressure on each other. And that pressure on the membrane literally triggers genes called tumor repression genes. And it tells the cell it's time to stop growing, right? Cancer is very oftentimes lacking that inhibition message, right? So cancer that just keeps growing and growing and growing causes tissues to no longer function properly. And that's the problem with cancer, is that the cells are growing uncontrollably. They steal nutrients, they steal space, they interfere with the function of that organ. And so when cancers insert themselves in those genes that would normally be triggered to say stop growing, then that's usually what we mean by interfering with tumor repression. I like to think of the growth factor, or the growth control process in a cell, think about like a vehicle. You have two ways that you can control the speed of that vehicle. You have the accelerator and you have the brake, right? Now, you're not supposed to have both of them on at the same time, but the accelerator would be the oncogene. It's the thing that says grow, grow, grow. The brake is the tumor repression gene. So if someone cuts the lines to the brake, you're going to grow, 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 and there's no way to stop, right? So that's the sort of the connection there. So we understand that viruses associated with cancer are sort of explained by what we call the oncogene theory, sometimes called the multiple hit theory. So here again is the normal state. Your DNA contains proto-oncogenes, which are essentially that accelerator. It tells cells to be in full-on grow mode during embryology. And then we have repressor genes that if they're active, they're producing proteins that are called repressors that inhibit that growth. And so under the normal conditions, cells get to their mature size and they stop growing and you don't get cancer. If you have a hit to the DNA, so here's a virus that, for example, has inserted itself next to that proto-oncogene, and it literally converts that oncogene into, or the proto-oncogene into an oncogene. It's sort of like pushing harder on the accelerator, right? Well, as long as the brake still works, as long as the repressor gene is still intact, it will inhibit the growth of that cell and you'll still have no cancer. The problem is if there's any second hit to the DNA at that repressor gene, now that cell is going to grow uncontrollably. And so that second hit, by the way, it doesn't even need to be a virus. It could be the other things that we think of as cancer causing, right? It could be radiation, could be UV, could be anything that interferes with the DNA or alters DNA. And so in some cases it is two different viruses that have a particular affinity for these particular genes in certain parts of the body. So for example, hepatitis. We know that the combination of hepatitis B and hepatitis D, that magical combination is a very high risk for liver cancer because those two viruses happen to have very high affinity for these two genes. And so why didn't we know this before? Well, number one, viruses tend to be contagious. Cancers were not understood to be contagious, and there's usually a very long time lag, right? We're talking about coming in contact with these two things over the course of your lifetime. You may not make the connection between the two, but the ones that we know for sure, things like Burkitt's lymphoma. This is a rare form of lymphoma that's associated with the Epstein-Barr virus. Some people may know this as the monovirus. Monovirus also, depending on when you're exposed to it, may lead to Hodgkin's lymphoma. And so remember, Hodgkin's lymphoma is basically cancerous B cells. And if those B cells are infected very early on, it actually protects the individual from Hodgkin's. If, on the other hand, your first exposure to the monovirus is in your 20s, it exponentially increases the risk for Hodgkin's later in life. And it's because of just how it sort of stimulates the immune system to either ignore or accept that virus, so to speak. Kaposi's sarcoma. This is that cancer of the blood cell or blood vessels that creates those lesions associated with HIV/AIDS patients. So I always think of that characteristic lesion on the forehead of Tom Hanks in that movie Philadelphia. That was the first outward sign that he had AIDS. And it's unfortunate because by the time this cancer kicks in, the HIV has really taken the immune system out of commission. So this is actually a combination of HIV and herpes 8. So it does require two different viruses to cause this condition. Cervical cancer. Of course, human papillomaviruses, there are hundreds of them, but we know there are 6 strains that are very likely to cause cervical cancer. And so we have a vaccine. We actually have a couple of vaccines. We'll talk a lot about vaccines when we get to further chapters down the road. But Gardasil was the first one that came out that protects at least the 6 common strains that are associated with the cervical cancer. All right, now shifting gears a little bit, we'll talk about prions, which we talked about earlier. We introduced in the first chapter. This is really a unique kind of infectious organism that is actually just a protein. So they get the name prions because they are proteinaceous infectious agents. So that's where the prefix there comes from. And essentially what they are is a single protein that got named protease-resistant proteins. So we have the PRP proteins. Think about what the word protease means. That's an enzyme. Proteases are very generalized enzymes that should be able to break any amino acid sequence up into individual amino acids. So proteases should be able to chop up virtually any protein, but these proteins are resistant. What their purpose is, now all mammals have a gene for PRPs. We have proteins in the cell membranes of our neurons, which are very long-lived cells. Remember that neurons are amitotic. They cannot repair themselves or replace themselves if they get damaged. And so because neurons have these long extensions, think axons, we have some axons that go from the brain all the way through the body and down to the muscles that move your toes. You have to have really stable membranes to have that kind of a structure. And so we have PRPs that help make those cell membranes more stable. So here's what they look like, right? So we have two basic tertiary structures in these proteins, right? We have primary structure is your amino acid sequence. Your secondary structure is just the folding patterns, and then tertiary is the hydrogen bonding pattern that comes from. And so we have what are called cellular PRPs. These are essentially the normal ones, and they are generally going to have more alpha helices, right, which is just one of the two ways we can fold these guys. Keep in mind sometimes that little C superscript, they sometimes refer to this as the common PRP as well as cellular. The prion, or the disease version of it, is where it gets misfolded into a beta pleated sheet rather than an alpha helix. So you'll see the PRP with a superscript P referring to the prion PRP. Prion PRPs are not replicated, right? They actually, if you get one of these in the body, it will move around the body until it bumps into those cellular PRPs, and it will convince them to reshape or change their shape into the beta pleated sheet, which basically is non-functional, and they will sort of congregate together. So whenever you have prion PRPs, they sort of migrate across the membrane, and they congregate together, and they make what they call plaques, vacuoles, and those vacuoles are due to the bunching pattern of these prions. And so once that happens, then the nerve cell that they are attached to is going to die. And so that's what causes the damage to the neurological system. It's not really well understood how they're transmitted, but what we do know is that they are ingested. So that is the primary route of transmission. There have been a few rare cases where organ transplants have transmitted this organism, but we know in humans primarily it's ingestion of contaminated meat, right? And so think mad cow disease, right? We were worried about it getting into the human population if people were eating contaminated meat. In wild animals, it's transmitted primarily through the environment. So if urine, saliva, so think about when we talk about CWD, or chronic wasting disease, in deer, right? There's a reason why there's a lot of hunting regulations now where you're not supposed to use salt licks and mineral blocks, things like that, where you're inviting animals to congregate together and share saliva, right? If they're swapping spit, they're swapping prions potentially. And so when those substances also get in the soil, it's very stable in the soil for up to 3 years. So we know that is mostly environmental transfer. In captive animals like the cattle that we think of as mad cow disease example back in Europe, it was through contaminated feed. And so why mad cow popped up originally in Europe is because that part of the world, the climate is not suitable for growing soy. So here in the US, what we feed our animals is a combination of corn and soy, because soy is a good protein source, right? And so in Europe, they can't grow soy. And so what they feed their cattle for protein is essentially all of the stuff that can't be used economically from sheep. And so the nervous system, the bones, all the internal organs, anything that's not harvested and marketed, they essentially mix together into a substance that they call sheep meal, right? And so basically they were feeding their cows contaminated brain matter from sheep. And so that was how it got into the cattle in the first place. And so it's unfortunate because now, of course, now that we understand that transmission, Europe imports a lot of soy, and because they don't want our genetically modified soy from the U.S., they're importing it from other countries like Brazil. And so because this became a big boom in economic exports for South America, now they're like chopping down the Amazon forest so they can grow more soy so they can make money off of Europe, because Europeans like their hamburgers just as much as we do. So there's a direct link between deforestation of the Amazon rainforest and Europeans liking cows or beef. So it's kind of unfortunate. But we also know that they can transmit it between animals in that contaminant in the soil, just like in the wild animals. There is some evidence that the type of soil that is involved also may help transmission. So places that have very low copper levels, that prion tends to be more readily spread. And so that's a little bit of information we have on the environmental aspect of it. So what do they cause, right? We know, in general, we know CWD and we know mad cow, but those are types of diseases that belongs to a family of diseases, what we call the transmissible spongiform encephalopathies. So TSEs are the prion diseases that we're talking about, and they all have similar manifestations in the body. They basically damage those neurons. Where the neurons are damaged, it's permanent. So you have this degeneration over time where you get these large, open, vacuous areas, what we call, makes it brain area looks very spongy, which is why it causes behavioral changes, right? And so where these things happen, it microscopically or histologically looks very similar to what they call plaques in Alzheimer's patients. And so Alzheimer's is based on what they call plaques and tangles, how the damage is manifested in the brain. And so because this looks very similar to plaques, there's a lot of crossover between Alzheimer's research and prion disease. There does not seem to be a correlation between the two, but because some of the tissue damage looks similar, there's some crossover there. And so it's a very slow but sure process. In fact, all TSEs that we know of are untreatable and fatal eventually. So again, it could take decades for the damage to actually take effect. But prion diseases are hard to deal with because the only thing that can damage or destroy a prion is incineration, right? So obviously that's how ingestion becomes a problem because people aren't eating ash, right? In order to kill that prion, you would have to literally destroy the substance that then gets eaten. So cooking is not effective, and that's why it gets transmitted in the food. Autoclaving by itself is not effective, and this is where it becomes a problem in medicine. So you can put it in an autoclave, but you have to add sodium hydroxide, which is a very powerful base. And so it's very impractical. Most things that might need to be autoclaved in the hospital couldn't sustain that exposure. You can't necessarily autoclave an entire surgical ward, so it becomes a real problem in medicine because we can't get rid of them if they've been introduced into a hospital setting. In farming, they have developed what's called Prionzyme, which is an enzyme that they can use if they discover a hot spot, or for example, all those fields where the mad cow cases were identified, they can use this Prionzyme to at least decontaminate the soils in those fields. But it's not something that's effective in the clinical settings, things like that. And it's not 100% effective, but it definitely reduces the concentration of prions and significantly. So it can help to sort of stem the tide of the problem if they can identify it. Because these things are untreatable, there is a lot of research into trying to find vaccines. It's still a very rare disease, but knowing that it's untreatable and fatal, there's some research that goes into it. Some of that research is tied to fungal research because they have actually found similar organisms in fungi, but obviously there's going to be a lot of differences. Fungi don't have nervous systems, so there's not a lot of correlation there. But they think if there's something happening at the cellular level that they can work out in the fungal prions, then they maybe be able to translate that into the animal version. So the examples of some of these diseases, the first one ever discovered was called scrapie, and they called it that because the neurological changes that happened in the sheep and the goats that were infected would cause them to literally scrape themselves up against the fences, and so the farmers would notice this behavioral change and they would cull that animal from the herd because it was clearly infected. Cows, so what everybody tends to call mad cow disease, its really fancy name is bovine spongiform encephalitis. In humans, right, there are some natural diseases that associate with humans. Now, humans can get mad cow disease by eating the uncooked beef, but humans can also get prion disease by eating each other, right? So Kuru was known as the laughing disease. So there's the behavioral change that happens in humans, and it's called Kuru because it was associated with a tribe out of South New Guinea where cannibalism was very common. And so these individuals would, there's some mourning rituals associated with if you were a warrior, right? If you were a man that was protecting the village, if one of these warriors died, the family would mourn the loss of that individual by cannibalizing their body. And so the village would, essentially, the women and children would be responsible for preparing the body. And you had to be an adult male to eat the sort of the manly parts of that carcass, the muscles, right? That's what made him a great warrior, were his muscles. And so the men got to eat muscle. The women and children got everything else, the brain, the organs, and the places that were contaminated. So how that adult male got infected in the first place, he had to have been exposed as a child to an infected spinal cord or something like that. And then as an adult, he then transmitted it on to the. So, of course, this is outlawed. It's been outlawed by the New Guinea government back in the 50s, but because this is such a long, there's such a lag time between it, and I'm sure even though it got outlawed in the 50s, it didn't stop happening immediately. And so we're still seeing cases of Kuru in some parts of the world because that ritual did not go away immediately. What's known as Creutzfeldt-Jakob disease, there's a genetic version of this, but sometimes you'll see an F in front of that for familial CJD. But the variant form of CJD is the form that is infectious, right? That's the one that would actually be the background of Kuru. That would be the one that you would get if you ingested infected animal tissues and things like that. Cervids would include those hooved organisms like elk and deer. So chronic wasting disease is maybe one that you've heard of because it is something that has become identified here in Arkansas as of 2015 was the first confirmed case. The Game and Fish has been testing for them since 1997 when they moved elk back into Arkansas from areas that are known to have CWD. But that's not necessarily how it got here. They believe it was probably here already. We just are now looking for it, so we recognize it and are testing for it. But even some other farmed species like mink, right? We farm mink for their coats, for their pelts. There is a form that is transmitted amongst them in their captive environments. Same thing goes for cats. Now, this is probably not your pet cat. These are going to be big cats that are found in wildlife preserves or zoos, places like that are captive, is where they've identified this. But clearly, they're capable of being infected by prions. So, and even in other captive situations, they have seen even other species. So when we say ungulates, we mean zebras and other kinds of grazing animals, but ostriches, right? That's the big shocker because ostriches are not mammals; they're birds. And so they suspect it's because of the unnatural situation where these animals are captive. So this is not in wild ostriches that they have identified this. It's only in captive species. So there's a lot of research into other species because clearly it's a pretty broad-ranging host options here. So just a little data here. If you look at as of 2016, this was the known distribution of the chronic wasting disease, at least in the U.S. and Canada. You can see clearly out west, it's much more prevalent. Arkansas now makes the records here. If we zoom in on Arkansas, you can see those initial cases were up in Newton County, and most of the cases are still up in that part of the country or of the state, but clearly, this is why there's been a big push for changes in hunting regulations because there's some evidence that shows if you have more than a 20% incidence in a population, it's no longer possible to eradicate the disease. All you can do at that point is focus on the management of the disease. So not real sure what our current percent incidence is in the state. But the goal of all the regulations that have been put in place over the last 10 years or so is really just to keep the number of infections low, minimize the spread of the organism, and hopefully, avoid contaminating new areas essentially. And so some of the rules that are in place, things like preventing or prohibiting feeding stations, right? If you have a mineral block out there, you're inviting them to congregate and you're inviting them to share saliva. So that's a much more likely transmission mode. Regulating where you can move the carcass. So if you hunt in one county, you may not be able to you're not allowed to move the intact carcass. You can take the meat off the bones and move the meat, but if you take the bones with you, clearly that carcass is going to have the leftover brain matter and spinal cord and those things that are potentially infected. And if you move the carcass and then bury it somewhere else, you've just introduced that prion to the soil. Or carnivores are going to come along and expose themselves and transmit it to other parts of the state. So, these regulations are in place to hopefully stem the tide and limit the spread. And finally, there's another group of organisms that are pretty unique, what we call viroids. Now, don't confuse this with the word we learned early on, which was virion, which is just a complete virus that's outside of its host. These are plant pathogens, and so these are not directly going to be harmful to humans, but they are definitely economically important. This family of organisms can cause severe economic devastation in the potato industry, cucumbers, avocados, lots of different organisms or different crops that can be affected by viroids. And so what they are basically just pieces of RNA. And so they look a lot like RNA viruses, but they don't have a capsid. And so they don't get packaged up and transmitted the same way that viruses do. That other viruses do. They actually have to be injected into a new plant host using an insect that has a perforating or mouth part, right? So if they have sucking mouth parts, they can get past the cell wall of a plant, and so that's how they get transmitted. They suspect that these little pieces of RNA are sort of evolutionarily left over from the post-transcriptional stages, right? So when you transcribe DNA, you make a little strand of RNA. Well, now that they know more about the human genome, that RNA that leaves the nucleus doesn't entirely go to the ribosome. Sometimes it gets a little bit of editing before it actually gets translated. And so basically what you end up with are tiny little pieces of RNA that sort of got clipped out of that original strand of messenger RNA. And so they're tying these little random pieces that they find in viroid infections back to pieces of messenger RNA that just got edited, basically. So these are sort of interesting pathogens in that regard. So keep in mind some of the animations that you can see regarding the viral life cycles. There is a DVD from an old textbook that I used to use on reserve in the library on campus. So if you don't have access to if you didn't hear this lecture in class, those animations couldn't play at the same time as the lecture here. So if you'd like to see some of those animations that might help you with understanding the life cycles, especially the animal virus life cycles, just go to the library. You can use your ID and check out that reference DVD while you're in the library, and hopefully that information will help you visualize some of the things we've talked about here today. So, all right, that's it.